Okay, hello everyone. I'm Marcos. I'm a postdoc researcher in Bjorn's laboratory. And today I'm going to talk about Epitope prediction. I hope everyone's enjoying so far the workshop, learning a lot. So I'm gonna briefly talk about this topic. I'm gonna start my presentation, talk about Epitope biology recap. So if you want to use the predictions on the B cells, you need to understand some terms that we are using. So what is like a epitope? What is an antigen? What is a paratope? If you don't know, there's no problem. I'm going to talk about them briefly so you can know a little more about that. After that, I'm going to start to talk about the predictions tools available on IEDB for the B cells epitopes. I'm going to jump into the linear sequence-based epitope. After that, I'm going to talk about the discontinuous ones, discontinuous 3D epitope predictions. And I don't have like any topic specific for the conditional antibody design, but I'm going to talk about them between those slides. So I'm going to see that I'm going to talk about docking, and for example, antibody protein docking. I'm going to talk about it. Structural modeling, I'm also going to talk about it, but I don't have like anything separate, like a separate topic for that. I'm going to talk between my presentation. So I'm going to see. So starting the visa recap. So do you know what it is? If you're not from structural biology, it's quite difficult to understand what is that. Like it's just like a lot of drowning, colorful. But I'm gonna try to explain to you. So let's just open the here laser point here. So here you have the antibody for a crystal, okay, and here you have the influenza hemagglutinin from influenza A virus. This protein is a trimer, so I have the H1, H2, H0, but you don't have the zero here. So the crystal is a dimer, but like it's but the trimer, but the protein has like another chain that is not being shown by here on this crystal. And here you have the antigen. So this HA1 is an antigen because it's the protein that is interacting with the antibody. So antigen is a protein that can trigger the immune response. It can be, for example, for antibody, can be, for example, for TCR. So every time that I talk about an antigen, it's a protein that can trigger the immune response. Inside of this antigen, you have the epitopes. So what is the epitope? Epitope is the smallest region from the antigen that can trigger the immune response. So here, for example, the epitopes that is colored by red over here are the amino acids that is interacting with the antibody. So this is the amino acids over here that is being recognized by the antibody, is triggering the response from that antibody. We also have the paratope. So what is that paratope? Paratope are these amino acids colored by blue over here. This is the amino acids from the antibody that is interacting with the epitope from the protein. So this region here, we have this this region on the antibodies. We have the what is called VDJ region on the paratope. This VDJ region is really useful. Like it's very important region from like we have this on antibodies also on TCR because you got all the variability that necessary for the interaction from the antibody to different kind of epitope. So imagine yourself, you have like probably millions of different epitopes, and you need to have like a variability from the antibody to recognize not every epitope, but at least some of them, a lot of them. So for that, you have this VDJ, which provides the mechanism necessary for the antibody recognizing different types of epitopes. And just zoom a little bit into the antigen part. We have this binding site of the antibody here. This is the epitope. And here you can see what we call it discontinuous epitope or conformational epitope. So what is that? It's not every amino acid from the epitope that is interacting with the antibody. it's just some of the amino acids is important for the interaction. Here, as you can see, the red ones are the amino acids from the protein that is interacting with the antibody, and the green one is not. So you can see here, like some red ones, some amino acid that is interacting with is surrounded by amino acids not interacting with the antibody. You only know this if you have the structural data, the conformational data of your protein. So, and to be, you can say like the two residues can be part of the same epitope. They need to be like close to each other. So we work with something like a cutoff about 6 angstrom of distance. So if the two residues, they are far from, like more far from each other, something like higher than 6 angstrom, the antibody cannot recognize this, both of them at the same time. So for example, here, imagine yourself. Here you have amino acid that is monogenic, so trigger is interactive with the antibody, so it triggers a response. And here, for example, we also have like amino acid that's also immunogenic. But they are so far away from each other, the same antibody cannot recognize both of them at the same time. So they probably would be like different epitopes, because the same antibody cannot interact with both of them at the same time. Here, another example. I have a different antigen. We have like a spermioglobulin. The epitope over here is like alpha helix. So the residue needs to be face off the antibody, so the antibody can interact with. So the residue from the antigen needs to be exposed, needs to be accessible to the antibody. So, because for example, if the amino acid is buried, the antibody cannot reach this amino acid, so this amino acid cannot be like interacting with the antibody. You only know this if you know the conformation data from your protein. If you don't have the conformation data, for example, I can try to create like an epitope, like I can synthesize an epitope, like a linear sequence from this part of the protein here, and the antibody gonna recognize this. But you don't know which amino acid is gonna be useful for that. You don't know which amino acid is gonna interact with the antibody. You're gonna treat this if you don't have the conformational data. You're gonna treat this as like a linear epitope. You know this linear epitope is immunogenic or not, because you can test it. You can see, okay, this epitope here is triggering the immune response, but you don't know which amino acid inside of this epitope is interacting with the antibody. You only know this if you have the conformational data. And also, if you work only with the linear sequence, you're gonna lose some kind of quite important information. As an example, here you have another a different chain. Okay, this chain is quite close to this epitope, and let's say this chain also it is immunogenic. So this part over here can interact with the same antibody that is part over here. So the same antibody can interact with both of them at the same time. But since if you don't have the conformation data, you're gonna line up the sequence. Their sequence is going to be very far away from each other, so you cannot predict those two different regions are going to trigger the same response from the same antibody, because in a linear sequence they're going to be far away from each other. But if you see as a conformational data, you can see they are quite close, so they can trigger the same antibody response. So more than 90% of B cells epitope are estimated to be discontinuous, because there's not amino acid inside of the epitope is interacting with the antibody. That's why we say like you have discontinuous different as a linear, and but you only know this if you have the conformation data, or they're going to treat us like as a linear sequence. So I know everyone like it's exciting to start using our tools. It's quite interesting. You have a lot of nice talking today, and but like you need to think about it. when to use the epitope prediction method. It's not always. And I'm gonna explain why. Reality is always better than a prediction. So if you have like the reality, you have the prediction, you're only gonna trust in the reality data. As an example, I could say, imagine yourself, you are in a room right now. There's no window in our room, and you want to take a step outside. You want to, as Nina is always saying, stretch your legs between those breaks. And like, okay, I don't have any windows. I don't know how's the weather outside. I'm gonna open like a forecast weather website. I'm gonna see the prediction, the prediction weather, and say, okay, it's sunny, it's hot, the birds are singing, blah blah blah. It's amazing. I'm gonna take a walk, and you just step outside of your house. There's like a massive storm, like hurricane, like cars flying, and you say, oh my god, like my prediction was wrong. The website was wrong. It's not like sunny. It's like there's a hurricane outside. So the reality is always right, and the prediction can be right, can be wrong. So the first thing, okay, I want to work with immunological data. The first thing we want to do is try to looking for epitope that is publishing in our IEDB. IEDB is huge. Like there's a lot of information. So if you want to work with a specific epitope, the first thing you're gonna looking for this epitope, if there's any like assay from in vitro or in vivo data showing that epitope is immunogenic or not. If you don't have any information about that that is published already, or for example, you have like antigen. Part of this antigen has some epitope that is published. You know, like okay, this part of the antigen is immunogenic, but this other part you don't know, don't have any kind of information if it's immunogenic or not. With that, you're going to start using the prediction method. So you're only going to use the prediction method if you don't have any published data available on, and of course, you can always check if it's available or not using IEDB. So this is the website. This is the B-cell predictions website. This is the main link. You can click over here and I'm going to open this print screen I'm showing to you. I'm going to talk about these three predictions here. So first one using the linear epitopes, DiscoTope, Ellipro using the 3D data from discontinuous epitopes. Starting with the linear epitope prediction, we have different methods to try to predict the linear epitopes. Most of them is using the physicochemical properties of the amino acid to try to predict where is the amino acid inside of the protein. Because as I said before, the antibody needs to reach the antibody needs to reach the amino acid from the protein. So if the amino acid is buried, it cannot be reachable by the antibody. So these calculations over here, these methods over here, gonna try to predict the position of the amino acid inside of the protein. So if the amino acid is in surface or not, okay, needs to be in surface to be predicted as a epitope. If not, it's going to predict as a no epitope. We have quite different calculations to do that. For example, you can use like a bit turn calculation, check if the amino acid is hydrophilic, hydrophobic. But all of them is using the trying to map the residues inside of the protein. It's not taking account if the residue or the epitope is immunogenic or not. And there's a problem because like we have some, for example, some amino acid that is exposed, but it's not triggering immune response. So some amino acid it's open, but it's not epitope, not triggers immune response. So this kind of method is informative, but it's not the best one. For linear sequence, you have some machine learning methods, which is better compared to the other one because for training the machine learning algorithm, we are using positive and negative training data sets that was published before. So we're using this kind of data of negative, positive epitopes combining with the physicochemical properties of the amino acid. And with that, have a robust analysis. So the prediction accuracy is optimized. We have on IEDB, we have this BPRED 2.0. It's not the only one available. have also a BC-pred, BC-pred, but which one is available on IEDB is this one. So you're gonna, if you want to start using the linear prediction sequence, you can click over here on the first link. You're gonna open this layout. I guess everyone here is already familiar with the layout because it's quite similar to others layout from other tools. So here you have this white box. You can paste the sequence of your protein. If you don't have the sequence, you can paste over here the UniProt code or the Swiss prot ID of that protein. And here, down here, you can select which method that you want to use to predict the epitope. If you want to know better about those methods, you can click on any link over here, on any blue link over here, and you're gonna open a help tab. So here you have all the information, a lot of information, for example, information about the method, information about the parameters, information about the scores of each method. So it's very useful if you want to learn more about the method. Okay, I have this kind of protein. I don't know which method I need to use. So take a look on this. This is very useful. And also, if you want to know more about a specific method, you can try to read the article. So you can click over these blue links over here. You're gonna open this reference tab. Now, here you have all the paper available describing that method, which is always good to read. It's always interesting to learn about that. So going back to the prediction tool, I selected for this example, RBD region for SARS-CoV-2 spike protein. I'm not using the entire spike protein because that protein is quite big, and the output would be messy to understand. So for like a training set, just to show you the output, I think it's better to reduce the protein just for a small portion. So I'm working with the RBD region. Well, of course, be my guest if you want to use the entire protein or different protein, we can we can test it. If you have no idea which method you're gonna use the IEDB recommended one, which one is bpred 2.0. This usually has the best results. You just click to submit. Gonna open the output. So here, for example, have the linear sequence of your protein. The algorithm is basically creating a window and sliding this window over your sequence, and it's selecting the fourth amino acid of this window and scoring every residue before and after this the fourth position. And if that is creating this score over here, this is a graph showing the position of that amino acid over the x-axis and the score over the y-axis. So here I have the green ones. It's predicted as like probably is a no epitope or very low chance to be epitope, and the yellow one has higher chance to be epitope. So possibly is a predicted epitope. You have your threshold over here. You can play with that if you want to. For example, okay, there's a lot of possible predicted epitopes over here. I want to reduce this number. So I can raise the threshold to 0.7. So this line over here is gonna be up over here, more or less. And you're gonna have only this region as predicted peptides. We also have the average. So the average score for the entire protein. Also have the minimum and maximum. And here have the table. So this table over here is only showing the same thing as the graph, but in a more readable way. So for example, here, have the number 9 here. Start this peptide here, this epitope here starts with the 151 and ends on 188. This region over here, which has the best score so far, which is probably is a epitope. And also we have here the predicted residue score. Here's the score for each amino acid from your protein. And you have this column over here. So if the score is higher than your threshold, so that's that amino acid is predicted to be as epitope, gonna have the E letter over here. So for each amino acid, you have the E. It means that amino acid is predicted as a epitope, a positive epitope. And of course, you have different methods. So it's always good to try to work with, play with those methods. So try to find a consensus. So open your sequence, try to use different methods and try to find a consensus using different methods. So for here, for example, I have these methods using the beta turn, the flexibility, and also the bpred, and all of them scored like a really high score on this region, which means that probably this region is a possible epitope one. So consensus is always good, and like try to find a consensus. It's good if you don't have like anything to support you. Using as methods as much as you can makes more sense. It's better to reliable. You can reliable more in your data and in your prediction data. So we tested all this linear sequence, and unfortunately, all of this linear sequence, no method gave us AUC above 0.6. If you're not familiar with that, so AUC is a method. It's a calculation that you can check if your prediction is good or not. So prediction has AUC close to 1. So 1 is the perfect score. So very close to 1 is really good. It's amazing. And for example, if your predictors has AUC around like 0.5, it means like your predictor is only flipping a coin. So you cannot trust the results at all. So 0.6 is not a good one. It's informative. For example, you can narrow down, you have like a big protein. You can narrow down some regions that possible could be like a epitope. And you can start to start with that regions, you know. It's still like, it's not, the results not that good, but like it still can be very informative. You can learn about that. You can start using this as a kickoff to your study. But okay, as I say, like linear ones is not that good. So what else you can do using the 3D data? So 3D data is always better than the linear ones. Unfortunately, we don't have a lot of 3D data available because we have major 3 methods to try to generate this kind of data. You have the X-ray crystallography, NMR, and electron microscopy. X-ray is still the gold one. It is the crystals generate like a high resolution crystal, which is really good. So we can map it. For example, you have your antigen and your antibody, crystallize it. You can map it, which residues from the antibodies is interact with the residues from your antigen. So you can map the epitopes from your antigen, which is really good. The problem that X-ray like usually takes 6 months to be done. It's a lot of time and also quite expensive. NMR generate different kind of data. So for us, it's not that it's quite difficult to use such kind of data. So usually don't use this kind of data. And I also have the electron microscopy, which is not that far away. That kind of data was only generated low resolution crystal, but right now they are getting better. So like right now we have like some good quality crystal available generated by electron microscopy. And it's good because like this technique is cheaper and faster. You can like have your crystal in 2 days, which is amazing. So how you can access those data? How you can find those data? For example, I have a protein. I want to start using the 3D data from that. Where I can find it? First, you can try to use the Scepter, as Dr. Paulo has showed before. You can use it, or for example, you can use the IEDB 3D export. So we have like more than 200 BCR assays 3D available. You can export it as a table and can try to work with that. But for using our tools, for using the DiscoTope Ellipro, you need the 3D file, the 3D coordinate file. That one is available on the PDB database, so protein database bank. So going to there, I'm going to show you after, but you can go there and try to find the protein, the crystal of a protein that I want to work with. And with that file, you can start to using your prediction tools to find for try to mapping predicted epitopes. So as I said before, here we're going to start looking for discontinuous epitope. On IEDB, you have available the DiscoTope Ellipro. Those are not the only one available, but it's quite difficult to us to implement new tools because usually have the license, which usually is quite expensive. We also have the problem with the implementation. And for usually the demands a lot of resources, computational power to run the tools, which makes everything more difficult for us. So that's why we don't have a lot of tools that can try to predict discontinuous epitopes. Of course, we are always trying new tools, always take a looking, but like that makes everything more difficult. We tried before this protein docking algorithm. We didn't get good results about that. That's why we don't have any kind of protein docking available on IEDB. So we're going to start using the DiscoTope. As you can see, to access the DiscoTope, you can click over this link. DiscoTope was trained using 75 X-ray structure, the first version of the tool. The second version of the tool was we using way more structure because there was more available at that time. And also we combined the mapped position. So we have the positive and negative training data set with the physicochemical properties of the amino acids. Combine those kind of data together, generate DiscoTope. So DiscoTope 1 has like AUC around 0.7. DiscoTope 2 has AUC score around 0.73, which is really good, it's better than the linear ones. Everyone compared to any kind of linear ones, that's results better. And how to use it? So for using the DiscoTope, you need a PDB. So you need the PDB ID, or you need the PDB file in your computer. Can be you can download it by the PDB website, or you can model the protein using a different tool and generate a PDB file. So you need the PDB. If your protein has more than one chain units, you need to select which chain you want to work with. For example, if it has 2 chains, you need to select just one. So we need to know which chain we want to predict the epitopes. But for example, I only have the sequence of the protein. I don't have any crystal, and I never found any crystal. So let's try to find out together. You just can open the PDB website, click on search, clicking in sequence search, advanced search. Here you have this white box. You can paste your sequence. You can try to find this algorithm. Is gonna try to do like a blast inside the PDB website. You're gonna try to find any crystal available that has that sequence that I'm looking for. For here, for example, you have one crystal available. Here you have some information about the crystal, the organism, some of the publication data, the authors. You can click over here to download the file, so you can download the FASTA sequence, also the PDB format. If you scroll down a little bit, you can have the more information about the chains, which is important for using the DiscoTope. Also have all the information about the molecule of the chains, the organism as well, the linear sequence compared to the reference protein from UniProt over here. So let's choose the one chain. So for example, we're gonna select the chain A. But okay, I tried to run for Blast on PDB. I tried to find a crystal available, but there's no crystal available for my protein. What else can I do? So you have different kind of modeling tools. If you can use the homology modeling, if you have like any protein available that's quite similar to your protein that you want to work with, or for example, you can use the B initial ones. If you never worked with that before, I suggest you choose AlphaFold, because AlphaFold, like, they are like a killing app right now. They got like a really good modeling score, and like everyone's on that area is quite impressive of that and starting using. So if you want to start it, if you don't have any crystal available, try to model using AlphaFold your protein. So now we have your protein, have your chain A, you select the version over here. IEDB suggests using the 2.0 version. So the scores, they are different. If you're using DiscoTope 1.1 and DiscoTope 2.0, the scores are different. So the first one is minus 7.7 is the default score. For the 2.0 is minus 3.7. But the sensitivity and the specificity about all of them is the same. Only the score is changed. So what the score means is the same, but the score value is different. So you need to pay attention to that. So I'm gonna use for this one, the DiscoTope 2. I'm gonna click submit. I'm gonna open this graph. I guess everyone's familiar already. So the green one is positive predictions. The red one is negative predictions. So this one is, the green one's possible regions that has epitope that can be recognized by the antibodies. Here you have a threshold to play around. You can save the prediction as a CSV file. You can click over here to see the result as a table view. Here, you have as a table a score of each amino acid available in your protein. The green one was the amino acid is classified as a positive predictions. So you can see the prediction over your in a 3D view. So this 3D viewer is a GA small 3D viewer gonna load your protein, and you can select which residue was classified as a good prediction, and you can click over here. You're gonna open this amino acid is gonna be highlighted, so you can see the position of that amino acid inside of your protein, which is also really good. You can say, okay, this makes sense. This is very exposed. So definitely, this is an area this epitope can be reached by the antibody. So it's always good to see the epitope, the prediction inside of your protein to see if it makes sense or not. And lastly, you have the Ellipro. So the Ellipro is good for linear and also discontinuous epitope predictions. It's using the Thornton's method. So this Thornton method takes the protein, try to transform that protein ellipsoid, and for each residue, it's going to calculate it how far that the amino acid protrudes out from the protein. So how far that amino acid is exposed. So with that calculation, generate like a index over here, call it like protrusion index. This is like a score. The method was quite old, like was published in '86, but doesn't have any tool. So we created a tool based on this method. Was published over here in 2008. So the layout is very similar to the other one. So here you have the same PDB ID, or you can upload your own PDB if you want to. Here's the minimal score. It's based on this PI score. So higher the threshold, fewer going to be the epitope that will be predicted. The maximum distance is longer distance. You can predict more amino acids inside of the same peptide, so inside of the same epitope. So that's calculation about like how far the amino acids can be from each other to be classified as the same epitope. Use all the information. We can use all as the default as well. Gonna select which chain we want to work with. We can work with both of them. Here, I'm gonna select just the first one, the A chain. Click submit again. Here, you have the predicted linear epitopes. Here, you have the predicted discontinuous epitopes. You can see those as in a 3D viewer. You can check the residue scores. Gonna open output like that. The yellow one is possible is highlight It's a high probability of being an epitope. The green one is low probability of being an epitope. I also have a score for each residues, and that score is based on the calculation from Ellipro. So basically, you can work with linear sequence and also confirmation data if you want to. It's always better to work with the confirmation data because the prediction works better, is more reliable, the results. And of course, every time it's like we're getting more and more tools available, we're gonna test it, more and more tools, and if you can, sooner or later, some of those new tools gonna be implemented on IEDB. And that's it. Thank you so much for your time.